Hi, my name is Lisa Manning, and I'm director of the Bioinspired Institute at Syracuse University. I'm also the Keenan Professor in the Department of Physics. So, the novel coronavirus has changed all of our lives in many challenging ways. In our institute, researchers have been working hard to find new ways to teach and train students despite social distancing, balance work and family obligations, and find creative ways to make progress on research that leads to new discoveries and positively impacts our society. One of the things I've been most impressed with during this difficult time is that a significant fraction of our researchers have pivoted to actively working on the novel coronavirus-related research. We all want to defeat this terrible disease, and some of us within Bioinspired have been able to extend our unique expertise to fight the virus. For example, Professor Allison Pattison, who was recently hired into the Bioinspired Institute and the Department of Physics, is spearheading a collaboration with another physics professor and Bioinspired member, Professor Jen Schwartz, as well as international collaborators. Their goal is to better understand how the novel coronavirus infects cells. Professor Pattison is an experimental biophysicist who studies the cytoskeleton. The cytoskeleton is like a cell skeleton. It provides internal support to the cells via a network of specialized proteins. And Professor Pattison is a world expert in how a special protein called vimentin alters the mechanics of the cytoskeleton. She's collaborating with Professor Jen Schwartz, who's an expert on theoretical modeling of the cytoskeleton, as well as theoretical models of the cell membrane, which is the surface that surrounds the cell. Although most of the work in the cytoskeleton focuses on its behavior inside the cell, previous work has shown that some of the cytoskeleton proteins, specifically vimentin, can be found outside the cell membrane. The amount of these molecules that end up on the outside of the cell have a strong impact on whether past versions of coronaviruses could get inside cells. So, Professors Pattison and Schwartz are pivoting to study whether external cytoskeletal molecules can help the novel coronavirus get inside cells. They think that the sticky vimentin molecules, which look like green hairs in the diagram, may help the virus, which is shown in red in the diagram, attach to the cell. All of this happens on the molecular scale, which is much, much smaller than the width of a human hair. The sticky vimentin molecules may even help the cell wrap around the virus, making it even easier for the virus to get inside cells. The movie that's playing shows a computer simulation of that type of interaction, and it was recently generated by Professor Schwartz's group. To complement those computer simulations, experiments in Professor Pattison's group will use state-of-the-art microscope imaging to measure the amount of sticky cytoskeleton molecules outside of the cell and study whether changes to those sticky molecules change the rates at which cells are infected by the virus. This can be analyzed using videos like this one, where virus-like particles are infecting cells, and you can count how many of the cells round up and die. This research project was very recently reviewed by other scientists who deemed it very promising, and so it just got funded by the National Science Foundation. These funds will allow our researchers to perform simulations and experiments and also train students at Syracuse in these state-of-the-art techniques. If Professors Pattison and Schwartz find that the protein vimentin does make the novel coronavirus more infectious, that is really exciting because it suggests that drugs that interfere with vimentin could be effective against the coronavirus. Since that's a very different attack point than many of the other drugs being studied, it would hold great promise for blocking the spread of coronavirus. A similar story comes from the lab of Professor James Hogland, who is on the executive committee of the Bioinspired Institute and is also a faculty member in the Department of Chemistry. So this is a picture of him and his lab. And he and his research group are also interested in the behavior of proteins near cell membranes at the surface of the cell. Their expertise is in pinpointing how the biochemistry of a protein, which generally speaking is how chemicals in a protein give it shape and function. So they want to know how that chemistry helps a protein live on the cell membrane at the edge of a cell. So the image on the left is a schematic representation of a complex protein. At one end of the protein, which is highlighted by that little box, you can see that there is a protein that has a biochemical change that allows the protein to interact much more strongly with the cell membrane. These specific types of biochemical changes that enhance the protein membrane interactions are very common in human disease, and Professor Haugland and his team are leading the world in those types of studies. So, when information about the coronavirus first came out, Professor Haugland reviewed the viral proteins to see if his research lab could contribute in some way to fighting coronavirus. A schematic picture of the virus is shown at the left, while a representation of the different proteins that make up the virus is shown in the diagram across the top. So fairly quickly, scientists figured out the roles of the proteins labeled in green, including the S spike protein, which you may have heard about already. The green ones are needed to form the virus and infect a cell, but the blue labeled ones are more mysterious. Although it's likely that they make the virus more effective at affecting organisms, we don't know what they do because the coronavirus is so new. Professor Haugland quickly realized that one of the blue labeled proteins, the one that's now circled in orange, shared remarkable properties with proteins he usually studies, and that suggests it could help the novel coronavirus interact with the cell membrane. He and collaborators in Georgia have been encouraged to submit a proposal to the National Institutes of Health to study this possibility. He plans to also investigate whether this type of protein membrane interaction helps the coronavirus to infect cells. If so, this is particularly exciting because we already have a class of drugs that have been studied in humans that target this specific type of protein membrane interaction. So Professor Haugland's work could potentially identify those drugs as a new route to attacking the coronavirus and blocking its spread. It is precisely these types of collaborative projects that require input across discipline boundaries and which value flexible thinking and openness to new ideas that drive us at the Bioinspired Institute. Our focus at the intersection of biology and material science has helped us to be uniquely positioned to address new health challenges. I am really proud to work with these researchers at Syracuse University who have, in less than 2 months, found creative ways to extend their existing tools and knowledge to address this global pandemic. Moreover, they're training a generation of students who will learn these cutting-edge techniques and be ready for the next challenge the world throws at us.